Okay, I want to thank everyone for coming and remind people to turn their cell phones off. And also remind you that after the lecture and questions today, there'll be a reception back beyond the elevators here for those who are interested in having a chat with Dr. Ntambi. James received his BS and MS degrees in biochemistry and microbiology at Makerere University in Kampala, Uganda. He then received a PhD in molecular biology at the Johns Hopkins University School of Medicine in Baltimore. He began his independent career as an assistant professor at Georgetown University in DC, where he stayed for a few years, and then he moved to the University of Wisconsin at Madison, where he rose through the ranks to become professor, and now he's an endowed Steenbock professor in the Department of Biochemistry and Nutrition at the University of Wisconsin. And actually, James and I both worked at Wisconsin. I was a graduate student there years before James got there. And it's a beautiful campus to work, although it's a little cold during the winter. In fact, I came from Tampa, Florida to go to Wisconsin, and my friends all thought I was crazy to go from the Sunshine State up there to study. There were other options. Well, James is even crazier than I, because he went from Africa to Wisconsin. James has received numerous awards throughout his career. He served on a number of NIH study sections and National Academy of Sciences committees. And he works in a very important area, an area that's increasingly important, and that is the area of polyunsaturated fatty acids. As you know, polyunsaturated fatty acids, like olive oil and fish oil, I think there's no question they're good for your health. And James works on an enzyme that actually produces unsaturated fatty acids in the body, and that is Stearoyl-CoA Desaturase, where he's really done some fantastic work that he'll discuss this afternoon. So the title of his talk is The Role of Stearoyl-CoA Desaturase in Metabolism, Implications in Human Diseases. James?
Yes. Thank you very much, Frank. I'm really very glad to be here at NIH. I can see faces of people I've collaborated with over the years. So I'm really glad that I'm going to talk about some of the work that we have done in Wisconsin for a number of years now. So just to get started, I just wanted to orient you to this metabolic chart that most of you are familiar with. It basically represents what a normal cell does. So at the cellular level, also at the organ level, and at the whole, organism level. And we believe that if everything is right, if there is homeostasis, then there is probably no disease control. But we also know that most of the environmental factors, most of the dietary factors, the hormonal factors, impact on a number of enzymes and genes that encode enzymes that catalyze most of those reactions that are shown here. So this metabolic chart is not really to impress you and to basically force you to understand everything on here. But we have chosen in our lab to do genetic regulation of metabolism, and particularly looking at enzymes of lipid and carbohydrate metabolism. So we think that inability to modulate lipid metabolism to maintain homeostasis is a hallmark of many human disease states, including metabolic syndrome or the obesity, diabetes, type 2 diabetes, cardiovascular disease, fatty liver disease, inflammation, atherosclerosis, and so many other conditions, including cancer. So we need to maintain homeostasis, as shown on that chart, to lead normal lives. So fatty acids, as you know, that flux in tissues, in tissue lipids, from either from the diet or from dietary sources, or are made de novo from glucose. And so this is basically the de novo pathway of fatty acids, starting from glucose through acetyl-CoA, and most of you are probably familiar with the enzymes of fatty acid synthesis until you get to palmitic acid, which is 16-0. And this now goes to the endoplasmic reticulum to make 18-0, which is stearic acid. Now, the enzyme that we are going to be talking about, Stearoyl-CoA Desaturase, takes these saturated fatty acids and introduces double bonds to form 16-1 N7 or 18-1 N9 that become incorporated into a number of tissue lipids that are shown down here. So as you can see, the enzyme will use the palmitic acid coming in from glucose as a source, but it can also, the fatty acids can also feed in this scheme directly coming from the diet. So the reaction that this enzyme catalyzes is shown here. First of all, the enzyme resides in the endoplasmic reticulum of cells. And it uses electrons coming from NADH or NADPH, and in cooperation or collaboration with a number of other proteins in the ER, cytochrome B5 reductase, which is a flavoprotein, cytochrome B5, which is iron-containing protein, and in the presence of molecular oxygen getting to the ER, the enzyme abstracts protons from positions 9 and 10 of a number of fatty acid-CoA substrates to introduce a double bond at this location, and in this case, in the synthesis of oleo-CoA or oleic acid. And as Frank said, this is, oleic acid is one of the major monounsaturated fatty acids in tissues. So this is the reaction that we've been studying for years. And I'm going to show you some of the things that this enzyme does. It is just a small part of that metabolic chart that I showed you. But what it does in its absence or in the presence will alter a number of metabolic pathways. Now, we started this work when I was at Hopkins, working with Dan Lane on fatty cell differentiation, which is shown here. These are pre-adipocytes, and when they differentiate into fat cells, they deposit these lipid droplets, which enlarge to form the large mature fat cells. So this process can be carried out in culture in 53A1 cells. Now, we found out that as these cells differentiate into fat cells, there is a dramatic increase in expression of a number of enzymes involved in lipid synthesis. And one of those enzymes is the Stearoyl-CoA Desaturase, which is actually shown here. This is a northern blot showing the expression of SCD in the mature fat cell compared to the pre-adipocyte. So there is this dramatic induction, and there are so many other enzymes that behave this way. So this was our first manipulation of the expression of this enzyme and the gene, showing its involvement in the formation of these lipid droplets. And in fact, if you analyze this lipid droplet, most of the fatty acids that you find in there are this 18-1 and 16-1 monounsaturated fatty acids. So it is pretty dramatic. So we also went ahead and looked at the expression of the gene in vivo in the mouse by doing some manipulations, hormonal manipulations. In this particular case, we show that the expression of the SCD is induced in the presence of insulin. So you make animals diabetic and then you inject insulin, and you can show a robust increase in the expression of the messenger RNA corresponding to the desaturase. We also treated or fed these animals diets that are rich in polyunsaturated fatty acids like triarachidonine and trilinolenin, or even DHA, which is fish oil. And we were able to show that the induction can be completely removed if you add these polyunsaturated fatty acids to the diet. So these omega-3 fatty acids, the fish oil, can downregulate the expression of this enzyme dramatically. So this is a manipulation, and again showing you that in response to high levels of insulin, you can induce the expression of this gene. We can also do a similar manipulation whereby we feed animals with high levels of fructose. This is, of course, corn syrup, and you can see again a dramatic expression of the SCD gene. And again, in the presence of polyunsaturated fatty acids, you can repress the expression of the gene. So there are agents that will induce expression, just like I showed in the case of the 3-3-1 differentiation, but there are also agents that can repress the expression of this gene. Now, you can also do a similar manipulation in humans, actually. It has been known for quite a while that humans don't carry out that much hepatic de novo synthesis, but in collaboration with Ron Krauss and Jack Russis in California, we are able to take a cohort of humans that were on a low carbohydrate diet and then were put on a high carbohydrate diet, just like I showed in the case of the rodent. And we can show that using the desaturation index, that is increased levels of 16.1 or 18.1 in the plasma, we can show a dramatic increase in expression of the SCD, or the Stearoyl-CoA Desaturase. So by dietary manipulations, you can induce the expression of this gene. Now, the gene, or the enzyme, is encoded for by several gene isoforms in the mouse. So in the mouse, there are 4 isoforms: SCD1, SCD2, SCD3, and SCD4. And all these genes reside on chromosome 19. So it is a cluster of genes on that chromosome. And now, because a human, there are 2 SCD genes that we know do exist. It's human SCD1 and human SCD5. And now human SCD1 is on chromosome 10, and human SCD5 is on chromosome 4. So these are not in a cluster. But apparently, structurally, these genes are very similar. Like mouse SCD1 is very similar to human SCD1, and human SCD5 is very similar to mouse SCD2. And I will show you the pattern of expression in a tissue-specific fashion. It turns out that the SCD1 is very abundantly expressed in lipogenic tissues, that is, tissues involved in fat synthesis or in lipogenesis, like white adipose tissue, brown adipose tissue, Hadarian gland. Some of these are very unusual glands, but they do exist in the mouse. And you can see the level of expression in the various tissues. So it is everywhere, but it's very highly expressed in the lipogenic tissue. SCD2 is also, like SCD1, expressed in these tissues, but apparently it is expressed in a more developmental fashion. SCD3 has a more limited expression in the Hadarian gland and in the skin, and a little bit in the perpetual gland of the mouse, whereas SCD4 is mainly expressed in the heart. So there is a tissue-specific expression of these genes. Now, the human SCD5 has a similar expression to that of mouse SCD2. Now, when we did manipulations initially, mainly in the mouse liver, we discovered that the expression of the delta 9 desaturase gene is highly regulated by a number of dietary and hormonal factors, as I told you at the beginning. And all these factors, like glucose, fructose, cholesterol, induce the expression of this gene in mouse liver. So is insulin. And you know, there is vitamins, vitamin D, some estrogen hormones, androgens, peroxisomal proliferators, metals, temperature. So you can see a range of dietary factors, hormonal factors, environmental factors, even drugs that we use in the treatment of certain diseases can impact on the expression of this gene. But at the same time, as shown in this column, we have molecules or agents that repress the transcription of this gene. So polyunsaturated fatty acids that I showed you, the omega-3s, leptin that we showed recently in collaboration with Jeff Friedman, can repress the transcription, thyroid hormone, glucagon, the TZDs, the compounds used in the treatment of diabetes, type 2 diabetes, repress. So you have a range of agents that induce the expression, and then you have agents that repress. So this led us to sort of investigate a little bit in more detail to figure out how these agents regulate the expression of this gene. It turns out that most of those agents that I showed you, either during induction or repression, they do it at the level of gene transcription. And most of, as you know, a number of transcription factors that have been shown to be responsive to dietary factors or hormonal factors. I'm showing you SRIBP1C, carbohydrate hormone response element binding protein, PGC1 beta, all these LXR, all these factors have been shown somehow to induce the transcription of the SCD gene in response to either high saturated fat or in response to high carbohydrate diets. Okay, so these factors are very important in the steps leading to de novo lipogenesis. And as you know, SRIBP is one of the master transcription factors that will turn on most of these lipogenesis genes. And then these fatty acids that are made here end up in these various tissue lipids. So because of that regulation, whereby you have a situation where some of these agents induce or lead to high expression of SCD, and you have this group leading to very low expression of SCD, this told us that maybe this enzyme has a role in metabolism whereby it controls or monitors the levels of some of the fatty acids that the animal receives in the diet, or it makes de novo. So at any particular time, based on your status, whether you are ingesting diets rich in these agents, you can induce expression and you have high levels. And when you have these agents in combination with these, you can modulate the expression of SCD along this line. And so that's what we think is happening in humans. So in order for us to understand in more detail the physiological importance of this enzyme, look at its role in vivo, we decided to use the opportunity of mouse genetics and try to knock out globally the SCD1 gene isoform. So we have made other knockouts as well, but I'll talk mainly about the SCD1. So when this mouse model was being constructed, was being made, there was another mouse model that had a natural mutation in the SCD1 gene, but that had a very big deletion. The mutation we made here is targeted to the SCD1 isoform. So the mice were produced, and as you can see, this is the mutant SCD1 minus mouse. This is the wild type. We have made the SCD2 as well. We have made SCD3, and we have made the SCD4, but those are not shown here. So the SCD1 immediately, it became very clear to us that it had a phenotype that had a dry skin and it had some eye defects. And we know the reason why. One of the reasons is that the sebaceous glands underneath the skin of these mice are deficient in the synthesis of cholesterol esters, wax esters, and triglycerides that are secreted onto the skin to lubricate the skin. So the eyes there's a gland within the eyes called the Meibomian gland that makes wax esters and cholesterol esters. And again, because the SCD1 is not there, they are not making enough of these lipids that would lubricate the eyelid so that the animal could blink properly. But in any case, in terms of metabolism, what we discovered is that the mouse, the mutant mouse, had very impressive phenotypes. So we were able to show that it had reduced, adiposity, and it also prevented liver steatosis, that is due to high fat or high carbohydrate diets, high steatosis due to OBOB, leptin deficient mouse model, PIPA alpha now mice, and the lipodystrophic mouse models. And you know, these mouse models tend to have high levels of lipid in their liver, so they have fatty livers. And we showed that if you put the SCD1 mutation in these models, you can prevent liver steatosis. These animals also exhibited increased insulin sensitivity, and here we are using the euglycemic hyperinsulinemic clamp experiment, and we are able to show that there is increased glucose uptake within this animal. It is resistant to diabetes, but it occurs in a leptin-dependent manner. They have increased energy expenditure, as shown by metabolic experiments. We put them in the cages and then measure oxygen consumption. They have increased thermogenesis because we know increased UCPs in various tissues, and also the beta-adrenergic receptor is highly expressed in the brown adipose tissue as well as in the white. They have increased fatty acid oxidation. And we know this because we have done microarrays in liver in particular, and we have shown that genes of fatty acid oxidation are upregulated. And very interestingly, this protein, MPK, which monitors energy status of the cell, is also upregulated in our mouse model. And of course, they also have reduced hepatic lipogenesis. And again, the mechanism seems to be a blockage in the processing of SREBP, the transcription factor that I told you. And there's also a blockage in the translocation of the carbohydrate response element binding protein into the nucleus to activate these genes. So all these phenotypes look very impressive, but we had a long way to show exactly what the mechanisms were in these tissues. But in any case, that was the global knockout. In other words, the SCD1 was knocked out in every tissue or in every organ of the animal. So now when we take these mice and we put them on a high-fat diet, and when we say high-fat diet, we are able to say specifically what those diets contain. Now, you always see when people publish papers talking about high-fat diets, they don't really specify. So we can show that in the case of a lard diet, which is very rich in monounsaturated fatty acids, or in coconut diets that are rich in saturated fat, when fed to these animals, they are resistant to high-fat diet-induced weight gain, as shown here in the green, while the white type are gaining weight, as you would expect. Now, what was even more interesting, that in addition to being resistant to high-fat diet-induced weight gain, these animals were extremely hyperphagic. This is one of the phenotypes that was very exciting, because you could actually see these animals in the cages eating as much as they could, and in fact eating even more than the ones that you are very familiar with, these OBOB leptin deficient mice. And of course, you know that the reason why these mice gain weight is because they are eating all the time. Now, our mice turned out that we are actually eating much more than the OBOB mice. And yet, they were not gaining weight when you put on a high-fat diet or even on a high-carbohydrate diet. And of course, we open them up. They remain lean, as shown here. These are the fat pads. These are epididymal fat pads. These are metal mice. And you can see they are small compared to the wild type. And even when we put them on a high-fat diet, they don't really increase that much in size. Whereas the wild-type mice, as you can see, increase, and the livers also become engorged with fat. So there is that resistance to high-fat diet-induced weight gain. So what is the mechanism? Now, one of the ways we would check whether these animals have protection was to introduce the mutations or the SCD1 mutation in various mouse models of obesity. So the OB mouse model is very well known, leptin deficient, hyperphagic, and is insulin resistant, and it is eating all the time. So it is a B6 background. There's this agouti mouse model, which is also obese. This one is interesting. It expresses leptin. It is hyperphagic. It is leptin and insulin resistant. So this mouse model is more akin to what happens in humans, whereas the OB mouse is leptin deficient, and there are very few humans in the world that are leptin deficient. And then you also have this diet-induced obesity mouse model. So we wanted to introduce our mutation in all of these to see whether they will be protected. And in the case of the OB, when we put the SCD1 mutation, we're able to show that, as shown here in green, there was a decrease in adiposity. So they decreased weight, and yet they were eating as much. And this decline or decrease in adiposity occurred both in males and females. Now, there were also increased lean body mass. There is also increased, so there's a decrease in fat mass, as shown here in green. There's also a slight increase in water mass. And as I indicated, this occurs both in males and females. Now, these models have one of the phenotypes that they exhibit, like the OB, they have a fatty liver. The agouti mouse model also have a fatty liver. So are the diet-induced weight gain. They also have fatty livers. And as these droplets are very clear when you do staining of the liver. So that's the histology of liver tissue. And all these are lipid droplets. So now when you put the SCD1 mutation in the OB, you can see a dramatic reduction in triglyceride levels. So is in the agouti mouse model, as shown here. So is in the diet-induced weight gain. And we have other models of increased fatty liver conditions. So this is a lipodystrophic mouse model. This is APO2 SRBP1C transgenic. And this animal is expressing SRBP1C in adipose tissue, but it becomes lipodystrophic and then deposits a lot of fat in liver tissue. So this is the partitioning of fat in tissues other than adipose tissue, and these animals become insulin resistant as a result. Now we put the SCD1 mutation, the liver tissue, the deposition of fat is decreased, and the animals become less insulin resistant. So we were able to show in collaboration with Jeff Friedman that one of the roles of SCD in the periphery is to mediate actions of leptin. So leptin, as you know, is made by adipose tissue, goes to the hypothalamus, works through the receptor, and then will mediate a number of metabolic pathways. And until this was described, there were not very many targets of leptin in the periphery. So we believe that some of the targets, or one of the targets of leptin in the peripheral tissues is SCD, and that it will mediate energy expenditure and some aspects of glucose and fat metabolism. So now in terms of insulin signaling, I remember I told you that the mouse is very insulin sensitive. Again, using the euglycemic hyperinsulinemic clamp experiment, we are able to show that there was increased glucose uptake in all these tissues, in the skeletal muscle, in the heart, in adipose tissue. And initially we thought that maybe the reason why these animals are insulin sensitive is because of their lineariness. As you can see, this is the mutant mouse, this is the wild type. But we are able to show that even the heterozygotes, that's the one that have only one copy of the SCD gene taken out, with the same weight and size as the wild type, again using the clamp experiment, we are also insulin sensitive. So the mutation is definitely causing increased glucose uptake in a number of these mouse models. And we had some insight in the mechanism of how this happens, because when we measure all the insulin signaling components, and this is even before you challenge the animals with insulin, we can see increased phosphorylation of the insulin receptor, the IRS1 or IRS2 and AKT, and of course we have measured increased glucose uptake. And one of the key players in this whole process was this PTB1B protein, which seemed to be downregulated in our mouse model. And we thought that maybe this, because of downregulation of this protein, there was a reduction in the removal of these phosphatic groups so that the insulin signaling pathway was continuously activated. But as I indicated to you, we also showed that there is increased MPK as another mechanism, and all these are tending to increased fatty acid oxidation when you increase the MPK. So in total, then what we found, and there are so many experiments that I didn't really describe here, what I want to say is that when we take out the SCD1 gene globally in the whole animal, we begin to see a number of phenotypes in the liver, in the muscle, in the brain adipose tissue. MPK is activated in the muscle and the liver. PTB1 is downregulated in the muscle. Beta-adrenergic receptor is upregulated in the brain adipose tissue. Now, if you pursue these, you see increased thermogenesis over here, increased insulin receptor phosphorylation, increased beta oxidation, downregulation of lipogenesis. And this, we think, is responsible for the decrease in let me just go back one slide here. So this is decrease in hepatic steatosis, increased insulin signaling, metabolic rate is increased, and all these together, put together, will decrease the symptoms of the metabolic syndrome and adiposity in general. Now, if you believe this effect of the SCD1 mutation on the MPK, and if you have been working on MPK, you know that MPK is a protein that really serves as a sensor of energy status of the cell, and it is involved in so many different processes. So you can see our SCD mutation can indirectly impact on a number of processes that are regulated by MPK. Now, we are also aware that in a global knockout mouse, every tissue is lacking the enzyme. So a lot of things can happen, but we know that in general, tissues talk to each other. So there is flux of metabolites from tissues, from the liver, from the adipose tissue, back and forth in the muscle. And we know this is happening all the time. And the idea is to maintain metabolic homeostasis. So I've just highlighted some of the key fatty acids that would be involved if you take out SCD1. And you have here 16-1N7 and 18-1N9. And of course, very recently, some Gohan at Harvard has shown that actually 16-1 can act as a lipokine that may modulate insulin signaling in some other tissues. So we are interested in looking at what happens to the various tissues, or what the contribution is for some of the tissues like liver and adipose tissue individually. So we have now sort of moved away a little bit from the global knockout of SCD, and now we are trying to look at the tissue-specific knockouts to try and sort of assess the contribution of each tissue to metabolic flux. And in order to do this, we use the SCD1 conditional knockout, and in the Cre recombinase, this is the Cre-Lox technology that you are familiar, some of you are really familiar with, where we have fluxed the exon 3 of the SCD1 gene, and then we take those conditional knockout mice, and then we cross them with the Cre recombinase driven by different tissue-specific promoters. And we can use the albumin Cre to knock out the SCD out of the liver. We can knock it out of the muscle using the creatine kinase. We have knocked it out of the skin using keratin 14. We have knocked the gene out of the brain using the nestin Cre. We have done the fatty acid binding protein, and we are going to use the adiponectin as well to knock it out of the adipose tissue. And we also have a way of knocking the SCD1 gene both out of the adipose tissue and in the liver simultaneously. And then you see how the cooperation between the liver and the adipose is. So we have done all these knockouts. And what I'm going to show you is some of the data that we have generated on the liver specific. So you remember when we fed the global knockout mice with the high-fat diets, and now I'm actually showing you the composition of some of these diets. So the ones shown in green are the coconut diet, and the high-fat diet, laddie diet, is the one shown here in brown. You can see the fatty acid composition of all these diets. So before we do an experiment, we have to analyze the composition of these diets to make sure we know what kind of fatty acids are in there. So I showed you that the global knockout was resistant to both high lard and high coconut and also high carbohydrate. So we have knocked out the SCD1 out of the liver, and we wanted to know whether the liver is really the key tissue that really helps this mouse to protect itself against high-fat diet-induced weight gain. Now, when we challenged both the lox, this would be like the control mice, and the liver-specific knockout mice with the lard and the high-carbohydrate diets, we got very interesting results. We found out that when we fed a high-fat diet, the animals were now no longer protected against high-fat diet-induced weight gain. But they were protected against high-carbohydrate diet-induced weight gain. And as you know, high-carbohydrate diets mainly induce de novo lipogenesis from glucose, and that's what I showed you at the beginning. Now, with the high-fat diet, laddie diet, we have monounsaturated fatty acids in there. So these go through the chylomicrons, they get into the liver, and they can actually replace whatever 18-1 the SCD1 would have made. So there is now a way of dissociating the effect of high carbohydrate from the effect of high-fat diet. So high-carbohydrate diet, in order to induce lipogenesis, requires SCD1. And we can show by RT-PCR that a number of genes involved in lipogenesis, like fatty acid synthase, acetyl-CoA-carboxylase, and elongase, are all downregulated in a liver-specific SCD knockout mouse. And that if in response to high carbohydrate diet, and in fact, if now we put 18-1 or triolein back into that diet, we can repair that, but we can't repair it with saturated fat like a steroid. Okay? So because there is no SCD, you cannot desaturate, and therefore steroid, instead of accumulating, actually gets catabolized. And this is basically showing you that lipogenesis using treated water incorporation in fatty acids is dramatically reduced, as shown here. And these are some of the enzymes of glycolysis. So in addition to downregulation of most of the lipogenic genes, we are able to show that the transcription factors that are involved in activating transcription, like SIBP1, as shown here, the maturation of SIBP1 is reduced, and also the translocation of the carbohydrate response element binding protein that would get into the nucleus to activate gene transcription is also downregulated. So we have an effect here in the liver where de novo lipogenesis is involved. So the liver actually is a tissue, key tissue, involved in de novo lipid synthesis. And in the absence of SCD1, that pathway is blocked. And the blockage is at the level of these transcription factors. We don't know exactly how that happens. And in fact, if you look at the liver steatosis now in the presence of high-fat diet, you don't really see any difference between the liver-specific and the logs. But in the presence of high-carbohydrate diet, you can see complete protection. So the high-carbohydrate diets contain a little bit of fat. We have to provide essential fatty acids so that the animal can survive. But we can now say that in the case of liver-specific mice, they are not protected from high-fat diet-induced obesity, and that the deletion of SCD1 in other tissues other than liver is probably required. Tissue like adipose tissue is required or necessary to cause sufficient decrease in the tissue and circulating monounsaturated fatty acids. And that liver-specific knockout will protect against high carbohydrate induced adiposity and liver steatosis. Okay? So the decrease in the nuclear levels of these transcription factors is probably part of the mechanism why these animals are not able to carry out de novo lipogenesis. Now, there's one interesting aspect to this. So we now decided, instead of using a high carbohydrate normal diet that we normally use to induce lipogenesis, we went a little bit further and used high sucrose diets that are devoid of fat, so that we could stress these animals, and the only way the animals can make fat is by de novo synthesis. Now, when we did this, we discovered that in the absence of no fat at all, the animals have defect in gluconeogenesis. So this is a pyruvate tolerance test where you inject pyruvate and then look for changes in glucose in plasma. And as shown here in green, you can see that the liver-specific knockout on a high sucrose, very low fat diet, they are unable to make fat. They have a defect in glucose synthesis. And these insulin signaling components actually are not really changing. So there is already a defect here. And we are interested in if, by the way, this defect can be corrected if we add 18-1 back to that high sucrose low fat diet, as shown here. So we can also look at other phenotypes that are exhibited by this mouse on a low fat diet, high sucrose composition. And one of the things that we observed is that if there is no fat and these animals are not able to carry out de novo synthesis, because SCD1 is not there, they develop a condition which is called cholestasis. And this is just analysis of plasma, and there is increased lipoprotein X, which is an indication of cholestasis, and there is increased bilirubin and bile acids in the plasma. And these conditions, again, can be corrected if you put 18-1 back in there. Now, in addition to that condition where we have no fat in the diet, and the animals are not able to make it, there are also other situations. And this is work that was done by Frank's group, Frank Gonzalez's group here at NCI, using this DSS. This is dextran sulfate sodium-induced inflammatory signaling model, whereby they are able to show that in the presence of this compound, there is, and probably by increasing bacterial flora in the small intestine, leading probably to either directly or to the production of these endotoxins, you can actually downregulate SCD1 in the liver. And what this sets in motion, now, so when you look at the expression of SCD in the liver, it's completely gone. So there is no expression, and these animals, I think, are on chow diet, and I have a feeling if they were put on a high carbohydrate diet, the situation would have been even worse. So there is SCD1 repression here, and when they do metabolomics, they see increased Stearoyl-CoA levels and in LPC, and they hypothesize, and probably they are right here, that this Stearoyl-LPC is now incorporated into these immune cells by the macrophages. So the macrophages now have more saturated fat in them. And this activates them, leading to cytokine production, the immune system here, as you can see, and leading to inflammation. So this is a model of inflammation and also colitis. So that is one example of SCD1 deficiency, complete removal in the liver under stressful conditions. Now, the work of Larry Rodell at Wake Forest, this is Mark Brown, they have also repeated some of these experiments using antisense inhibition of SCD and looking at macrophages. And they also are able to show that if they inhibit SCD1 in the liver, they can increase saturated fatty acid levels in the VLDL And of course, this will get into LDL and then transported in plasma. And this will get into the macrophages, activating the toll-like receptor 4, leading to cytokine production and inflammation. So what is this beginning to tell us? So what we think is happening is that in order for normal metabolism, you need oleate homeostasis. So we think that if you turn on the SCD1 gene, let's say in response to high carbohydrate diets under normal conditions, or in response to insulin, response to cholesterol, that will lead to these phenotypes. And if you now go very far all the way here, and you remove the SCD1, whereby the animal is not able to make monounsaturated fatty acids, you get into some of these conditions like inflammation and atherosclerosis or colitis. So this basically is suggesting that you can dissociate some of these disease conditions. So we remember that the metabolic syndrome, you have the obesity, you have type 2 diabetes, you have fatty liver, you have inflammation, and all these conditions are being lumped together. But in this mouse model, you can actually dissociate. We know that if you inhibit SCD, you can actually reduce obesity, you can reduce type 2 diabetes, you can reduce the fatty liver. But if you now go further and remove the capacity for the animal to make oleic acid, then you can now induce these disease conditions. So basically then, in normal circumstances, or under normal circumstances, we think that in human beings, if you look at this triangle, we think we lie somewhere on this line. That some of us have high levels of SCD expression, some of us have low levels of expression, and these dietary conditions, hormonal factors, can modulate the expression of the gene depending on what you are ingesting, depending on where you are in the world, environmental conditions, factors impacting on the expression of this gene. In some cases, you have insulin resistance with diabetes if you have very high SCD expression, and in case of low expression, you have fat burning, leanness in response to this omega-3, leptin, T3, and you have increased insulin sensitivity. And when you have almost no SCD, which is very unlikely, and so it is some kind of titration of the levels of expression of the enzyme. And of course, somewhere in the middle here, this will be the heterozygote situation in our SCD1 deficient mouse. So this will be the heterozygote. You can see here, if you stayed in this range, you are probably okay. If you are here, you can develop atherosclerosis inflammation. If you are here, you have these disease conditions. So this is the research group that has been involved over the years. It is a dynamic research group. It turns over. And they have really put in a lot of effort to characterize this enzyme, which initially we thought was a housekeeping enzyme, but has turned out to act as a molecular switch to enhance fat oxidation in the substance and sort of prevent fat storage. And I also would like to acknowledge a number of my collaborators. Some of them are here at NIH. Frank, George Connors. I've had opportunity to work with Alan Ari, who is in my department, Jeff Friedman at Rockefeller, Ronnie Kahn at Harvard, Ronnie Krauss and Jack Russes in California, and Jeffrey Peters at Penn State, and Matt Hoover at Virginia Tech. And of course, I wouldn't have been able to conduct this research if I didn't have funding from NIH and from American Heart Association and the USDA. And I would like to end here and again show you that we are very far off understanding what really goes on in the entire organism. We need to really go back and understand metabolic pathways. And I would advocate that although at some point some people were thinking that metabolism is over, that everyone now understands how this metabolic chart works, but that's not really the case. I've just shown you that by inhibiting or by sort of knocking out only one enzyme in one small area of the metabolic chart, you can alter metabolism in other places. And of course, other investigators have knocked out other genes in other different pathways, and they get different phenotypes. So I would suggest that metabolism should be brought back into the arena. And I will stop here and entertain questions.
We have time for a few questions. James, you certainly convinced me that SCD1 is important.
Yeah, right.
Yeah. So the SCD1 knockouts can overeat, but they don't gain weight. Can all of that be explained by the increased thermogenesis, or do they have a difference in activity?
We haven't really measured activity seriously. In fact, we are sending these mice to someone who can do those measurements. We don't have the facilities to do that. But when we just look at them visually, they look a little bit more active. But definitely the increased thermogenesis is well established. They have increased uncoupling as well. Yeah.
Beautiful results, Jim. The dramatic phenotype of the SCD1 knockout would suggest to me that compensation by upregulation, for example, of SCD2 is not really very effective.
No.
Now, do they upregulate SCD2, the SCD1 knockouts?
No, they don't.
They don't, okay. Because I thought that if they do, that would further indicate, since SCD2 is expressed in other tissues, that the monounsaturated fatty acids act locally and they don't have really hormonal-like effect once they're released from the tissue.
Yeah, so in the case of the liver, for instance, we found out that SCD2 is not really expressed in the adult mouse. Even when you challenge with a high carbohydrate diet, the SCD2 is expressed only in the neonates. And upon weaning, its expression is completely shut down. So it plays a major role in development, and then the SCD1 takes over and controls MUFA synthesis in the adult animal.
I have a couple questions.
Yeah.
Are there any human genetic deficiencies in SCD1 that you are aware of?
No, not anywhere I'm aware of. There are some polymorphisms that have been detected, but I haven't. They don't really give dramatic phenotypes. They are phenotypes, and in fact, they have shown correlation with body mass index in humans. In the work that Marty Hoover did in the obese individuals in the muscle, he showed that SCD1 expression by using microarrays, that SCD1 expression is very high, and compared to lean individuals. So there's this correlation of adiposity and insulin resistance when you have increased SCD1 expression. At least even in the tissue culture of human skeletal muscle cells, you can see the same thing if they are isolated from obese individuals.
My second question is SCD1 being pursued as a drug target?
SCD1? Yeah. Yes.
One more question. Is the body temperature of the SCD1 mutant high enough that it might cause brain damage?
No.
Okay.
These animals, we have had these animals for months, and they are not going anywhere. So we don't even know whether they live. I mean, their longevity. I mean, they have lifespan is increased. That is being measured right now. No, they do okay on a chow diet.
Okay, if there's no further questions, again, we have a reception right past the elevators on the right. Anyone that wants to talk about the mountain.
All right. Yeah. Thank you.